Hello, my name is Vasundhara Agrawal, and I'm the Applications Manager for Spatial Bioimage Analysis at Leica Microsystems. Today, I would like to go over our collaborative effort with cell signaling technology to map the cancer immune landscape in colon adenocarcinoma, or CAC, using multiplexed imaging and AI-based analysis. In this study, we visualized nearly 30 biomarkers in the CAC tissue using cell dive multiplexed imaging workflow and validated antibodies from cell signaling technology. Using this approach, we were able to probe multiple pathways of interest in cancer progression, such as vascularization of tumor tissue, immune cell responses, and cell proliferation, among other key molecular players within the tumor microenvironment. First, let's take a look at Table 1, which highlights the panel of CST immuno-oncology antibodies used in the study. The CAC tissue was imaged on the cell dive imager using 4 channels plus DAPI with automatic autofluorescence removal, corrections, and stitching. Now, moving on to Figure 1, uh, the fully stitched cell dive multiplexed images shown in Figure 1A were then imported and analyzed using Aivia. Next, moving to Panel B, the cell dive image was utilized to perform cell membrane and nuclear membrane segmentation to identify individual cells using a multiplexed cell detection recipe on Avia, followed by phenotyping to assign phenotypic markers to individual cells. Now, moving towards Figure 2, using AI-driven analysis, the expression of markers previously shown in Table 1 was characterized through clustering and dimensionality reduction methods. AI-guided analysis reveals single cells characterized by expression of multiple markers for epithelial, mesenchymal, and immune cell types. The first part of the analysis, shown in Figure 2A, utilizes unsupervised clustering using Avia analysis software. Clustering analysis showed a diverse range of lymphoid cell populations with activated T cell population, B cells, natural killer cells, shown in pink boxes in the figure. Larger groups of clusters specific to cellular metabolism, shown in blue, and proliferation, shown in orange, that are possibly indicative of tumor progression, were also identified. Within the myeloid cell population, specifically, tumor-associated macrophages, or TAMs, shown in green, were closely linked with endothelial cell populations, shown in red. And specifically, TAMs are characterized by their ability to facilitate tumor progression by supporting angiogenesis and preferentially accumulating around blood vessels. And in Figure 2b, we noted the spatial positioning of TAMs marked by heterogeneous expression of CD68 positive and CD163 positive cells close to the blood vessels in CSC tissue. Next, dimensionality reduction using UMAP in Figure 2c allowed us to visualize cell phenotypes within the heterogeneous CAC tissue and broadly clustered them into groups associated with tumor progression, innate, and adaptive immune cell populations within the stroma. Next, looking at Figure 2D, we can also appreciate how these distinct cell populations or phenotypes are spatially positioned within the whole colon adenocarcinoma tissue and highlight the complex relationships that exist within the tumor microenvironment. Together, these data allow the creation of a spatial map of tumor cell aggression and offer predictive value for the progression of the disease, and also interrogate the heterogeneity within the tumor microenvironment. Finally, if you'd like to take a closer look at the poster, it is available for download. Thank you for listening.